Okay, so this is where it all starts to get a bit more exciting. So we're going to talk about the innate response and the processes within that. We're going to have cells diving between endothelial cells into the tissues, phagocytosis, and right at the end we'll activate the adaptive response that we'll talk about in part 3. But before we get into all of that, we need to talk about some cells. And these are the cells that are producing or a lot of the chemicals that we were talking about before. Bear in mind we also have endothelial cells producing a lot of the chemical mediators of inflammation, the humoral component of the innate response. In terms of the cellular component, we are talking about leukocytes, white cells, and we have a few of them. So in the innate response, we have granulocytes. Remember, we talked about the chemical mediators being formed in granules in specialist vesicles within granulocytes. Well, these are the granulocytes. So under this, we have mast cells. Remember, they release histamine, serotonin, heparin. We have neutrophils. We have eosinophils and basophils. We also have professional phagocytes. They're called professional phagocytes because that's their primary role. And in these we can include dendritic cells and also the monocytes that become macrophages when they get into the tissues. Actually, some of the monocytes can become dendritic cells as well once they get into the tissues. And natural killer cells. Now, natural killer cells are a bit odd in this context because although in the way that we're going to talk about them, they're operating within the innate response, in other words, they're not antigen specific, they are actually lymphocytes, just like the T and B cells. Which brings us to the adaptive response. So here we have lymphocytes. Lymphocytes, if you remember, are a subset of leukocytes. They are found in lymphoid tissue. So we have T cells or T lymphocytes. Of these, we can have two flavors, if you like. We can have T helper cells or T helper lymphocytes, TH, or cytotoxic T cells, TC cells, both with different roles within the adaptive response. In the adaptive response, we also have B cells. Now, in terms of the adaptive response, often the T cells are thought of as the cellular component, and the B cells are thought of as the humoral component, the chemical component. And this is because once they're activated, they differentiate, they change, and they become plasma cells. When they become plasma cells, they secrete antibodies, little plasma membrane proteins known as immunoglobulins. Remember, a globulin is a plasma protein. Immuno is obvious because they're involved in an immunological response. So antibodies, and in some cases the B cells, are actually considered to be the humoral or chemical component of the adaptive response. So let's talk in a bit more detail about each of those categories. So we have our granulocytes. As we said, this is mast cells, basophils and eosinophils, Also, neutrophils come into this. We'll talk about those in a little while. Now, mast cells are found near all the external barriers, and this is why they're quite significant in terms of allergic responses, in terms of urticaria, in terms of bronchospasm, because they are very close to the skin, they're very close to the mucosa. They're also found near blood vessels. So basophils and eosinophils, though, tend to circulate. In order to activate a mast cell, the activation actually results in them degranulating. So they release their granules into the extracellular space. And they also secrete cytokines. So we've got degranulation of these preformed chemicals, but we've also got the synthesis and the secretion of our chemicals that are formed at the time. So they can be activated by binding directly to pathogens or to infected cells. They can also be activated by complement. Remember complement? Complement is one of our plasma-based mediators that's synthesized as C1 by the liver, then circulates in inactive form. It becomes activated, and if you remember, we go through the classical pathway, we get C3, we go into the alternative pathway, and we get these all these different complement proteins with different roles. So C3A and C5A, if you remember, degranulate mast cells. So this complement binds to the mast cells and the mast cells degranulate. Mast cells can also be activated by direct injury or infection, and by cytokines, in particular interleukin-1 and interleukin-8, and this binds to specific receptors on the mast cell. They can also be activated by neuropeptides. Neuropeptides, they come from our C fibers. They come from neurons. When certain neuron C fibers are stimulated, they release neuropeptides that help to upregulate inflammation and sensitize pain receptors, nociceptors. One of the key ones is substance P. Now, substance P, its role is to bind to mast cell receptors and cause the mast cells to degranulate, which of course releases histamine, which is a vasoactive amine. So indirectly, they cause vasodilation and increase vasopermeability. The other thing, and this is the main thing that you'll probably see in books, because this is the allergic reaction stimulus really. Mast cells and basophils carry receptors for immunoglobulins, for antibodies. And if you remember, antibodies bind to cells via the Fc end of the antibody, the bottom of that Y shape. So these receptors are known as Fc receptors or FCRs, and they coat the mast cells. When we release immunoglobulin that is specific for an allergen, those antibodies, those immunoglobulins, will bind via the Fc, the fragment constant end, the bottom of the Y, to the mast cells, to the Fc receptors. And this is where we get antigen-specific immunoglobulins. Antigen-specific immunoglobulins are synthesized and secreted by activated B cells or B lymphocytes that have differentiated into plasma cells. These plasma cells then secrete lots and lots of antibodies. Those antibodies, the top of the Y, the 2 points at the top of the Y, are antigen specific. The bottom of the Y is the Fc end, and it's the Fc end that binds to these receptors. In allergic responses, we're primarily talking about the immunoglobulin IgE. So when the antigen binds to these specific fragment variable or Fv ends and cross-links 2 antibodies that are bound to the mast cell, this stimulates the mast cell to degranulate. And this is where we get allergic responses, because we've already produced IgE. The Fv ends of these IgEs are allergen specific. They then bind to mast cells. Mast cells, as we know, are found near the skin, near the mucosa, near the blood vessels, all over the body. And they're coated with this allergen or antigen specific IgE. And this is why we can sensitize to an allergen. And then the next time we come across that allergen, we have a massive response to it, because our allergen is antigenic, it's the antigen, and it's binding and cross-binding these IgE on mast cells. But other than that, mast cells are really significant in terms of the innate immune response, because they are found throughout the body, and because there are so many ways of activating them. They are an early source of all the major pre-synthesized and newly synthesized mediators. Remember, not only do they degranulate, they also secrete newly synthesized mediators. Pre-synthesized in granules, of course, it includes histamine, heparin, small amounts of bradykinin, and serotonin. Newly synthesized is cytokines, particularly TNF-alpha, who I always refer to as our big bad boy. Chemokines, remember chemokines, they create this chemotactic gradient. They're responsible for chemotaxis, for guiding any new leukocytes into the source of the problem. Also prostaglandins. Remember, prostaglandins come from the arachidonic acid cascade, from the COX pathway. And proteases. Proteases are enzymes that break down proteins. So mast cells will release this whole cacophony of inflammatory mediators when they've been activated. Our other important granulocyte are neutrophils. Now, neutrophils may also be called, and you may actually see them in scientific texts, as PMNs. This stands for polymorphonuclear leukocytes. And this is because of the shape of their nucleus. They don't have a nice little round nucleus. They have this divided up nucleus joined by bits of chromatin. So they have a really funky, bulgy shape of nucleus. The mechanisms that degranulate neutrophils are not really fully understood. There are some theories and there are some mechanisms that have been proven, but others haven't, or they're inconclusive. But when they are degranulated, they release granules of proteases, so those enzymes that break down proteins, and lysozymes. Remember, these are our toxic chemicals that includes reactive oxygen, it includes proteases, it includes lots of chemicals that will break things down. They're also a phagocyte, and technically they're the most abundant phagocyte, but they don't get much chance to do it because they're very short-lived, and they tend to die when they do phagocytose. So it's a bit like bumblebees, really. It's alleged that when they sting you, they die. In the terms of neutrophils, they often die when they phagocytose or die when they degranulate. If neither of those happen, they will only survive 4 to 5 days anyway, and after then they become senescent. If they become senescent, they go into apoptosis and die. So they're very short-lived. They don't make good antigen-presenting cells because they are short-lived, and because when they phagocytose, they tend to die. One of the funky things they do, though, is they produce these neutrophil extracellular traps. And whoever named these was very clever, because of course that spells net. And they are like nets. So they use that nucleus that's all funny shapes, and they throw out chromatin. Within that chromatin, they have embedded proteases. So the chromatin acts like a net, it traps pathogens, and the proteases then start to break down the pathogens. Interesting thing about neutrophils is that they also produce an enzyme and release an enzyme when they die or when they degranulate called myeloperoxidase, MPO. Myeloperoxidase is green, and this is the thing that colours sputum in small amounts yellow, in larger amounts green. It's myeloperoxidase from neutrophils. Which brings us on to our phagocytes, our professional phagocytes, our ones that are really good at it. And the best ones at it are dendritic cells. And looking at the shape of the dendritic cell, you can see why. You can see why it's called a dendritic cell. Remember the dendrites on a neuron? This has lots of arms sticking out. Those lots of arms have lots of receptors, pattern recognition receptors, all the rest, to bind pathogens. So they can bind multiple pathogens at once, or multiple bits of dead host tissue or dead proteins. So they're generally found around the skin and the mucosa, and following phagocytosis, these frequently become antigen-presenting cells. These become the link between the innate response and the adaptive response. Our other professional phagocytes are monocytes that become macrophages. So we have circulating monocytes. They're released from the bone marrow as monocytes in response to colony stimulating factors. And once they get into the tissues out of the blood, they become larger macrophages. And then they start to function as phagocytes. When they do, they secrete many inflammatory mediators, so they are calling for help. They are causing leukocytosis, they are causing more phagocytes, more leukocytes of every kind to come and help them. Some of the macrophages that are routinely released will eventually, most macrophages will survive in the circulation for about a month. After that, they either die or they become tissue macrophages. So most tissues have their own resident macrophages. They've already got into the tissue, already converted into macrophages. And there they can survive for months in case they're needed. Good examples of these are Kupffer cells in the liver, histiocytes, they're found in the subcutaneous tissues, and you'll remember microglia in the central nervous system, one of our glial cells. And also right at the end we'll talk about alveolar macrophages. So obviously they're in a prime position there to respond if there's an invasion of pathogens, or respond if there's any tissue death, any cell death. They will keep it nice and cleaned up like little hoovers going around the tissues. The remainder are often known as wandering monocytes that then only go into tissues where there is an immediate problem, where they've been called to that area. Lymphocytes we really should be talking about, and we will talk about in the next part, because these are a subset of leukocytes, and generally we're talking about T and B cells stored in lymph glands but also in lymphoid tissue. So in terms of lymphoid tissue, we have skin associated lymphoid tissue, remember salt. We have mucosa associated lymphoid tissue, MALT. Let you into a secret, we'll also in clinical reasoning talk about gut associated lymphoid tissue, or GALT. So anywhere we have lymphoid tissue, we will have lymphocytes. Generally we're talking about T and B cells. So these are lymphocytes that are involved in the adaptive immune response. And they're activated in response to specific antigen presented by antigen presenting cells. So our dendritic cell or our macrophage will present the antigen to our lymphocytes and say, oh, come and sort this out, mate. We need you now. We need an adaptive response now to give us immunity. Natural killer cells, as I said, are also lymphocytes, but generally they function in the innate response and they are not antigen specific. They can be made antigen specific, and we'll talk about that in our final part when we talk about some of the functions of antibodies. In the innate response, they cause cytolysis of any pathogen or a damaged or infected host cell that is not carrying host MHC1. Remember, major histocompatibility complex. And remember, all cells, apart from erythrocytes, have MHC1. So those, if you remember, were our markers of self. We put up this MHC1 flagpole, and in it our flag says, yes, we're happy and healthy, here's some healthy proteins, we're okay, Jack. The natural killer cells aren't that interested in what's in the MHC1. They're not interested in the flag. What they're interested in is the flag post. And remember, we said MHC is unique to all individuals apart from twins. So they need to know that that is your host MHC. If it's not, then they can attack. When they attack, they exocytose something called perforins and granzymes. These granzymes are basically a type of protease. The perforins, they were so called because it was used to be believed that they made holes in the membrane, but actually it's now thought that they do disrupt the membrane, but what they're doing is assisting the entry of the granzymes. Once the granzymes get in, they trigger what's called the death pathway. They trigger apoptosis, which is cell death. So if a cell isn't displaying MHC1, they will bind, they will release perforins and granzymes, and that cell will then die. So let's have a look at that. So here on the left you can see a natural killer cell, sometimes abbreviated to NKC. In this case, it's found a target cell, the pink one. It's bound, and these always remind me a bit like Daleks, because they've got two things sticking out. They've bound to the target cell with one receptor. And then they have another receptor that needs to bind to MHC1. In this case, it's found MHC1, that MHC1 is expected host cell MHC1. That's absolutely fine. Again, it's not interested in what the MHC1 is displaying. It's interested in the flag post rather than the flag. But it's quite happy with this. This cell has MHC1. However, in this case, what we've done is the receptor on the natural killer cell has bound to the target cell, but its receptor that's looking for MHC1 hasn't found it. So that causes it to then degranulate. When it degranulates, it releases its perforins and granzymes, and that pink target cell there will then die. It will go into apoptosis, cell death. Of course, if it's an infected cell, any pathogen within it will also be contained and will be killed. So let's have a bit of a talk about cell death, because we have sort of two versions of cell death. And you remember we talked about membrane attack complex. We talked about it making pores in pathogens or host cells. Sodium floods in, water follows, the cell swells and eventually bursts and goes into lysis. But of course, when it bursts, it's going to release all those nasty toxic chemicals in the lysosomes. It's also going to release any pathogens within the cell. So it's not an ideal way to kill a cell. It's a messy cell death, known as lysis or cytolysis. So remember, MAC makes a pore in the plasma membrane, sodium and water influx, the cell swells, the cell finally bursts, and then, of course, as we said, this potentially releases lysosomal enzymes, which are nasty toxic chemicals, proteases. They will start breaking down our healthy cells and also the extracellular matrix. So they're going to make a bit of a mess when they're released. It also releases any intracellular pathogens. So those pathogens, for example, a virus, can go on and infect another cell. Also, it leaves us with a mess. It leaves us with a lot of necrotic tissue, a lot of broken proteins, broken cell, a lot of mess and debris. And that mess and debris then has to be cleared away by macrophages or dendritic cells before we can resolve that problem, that area of inflammation. So much more preferable really is apoptosis. So this is an example of this would be a natural killer cell, but in part 3 we'll also look at cytotoxic T cells. They do the same thing. So apoptosis is a clean death, because what happens in terms of natural killer cells and cytotoxic T cells, perforins and granzymes enter the cell. The granzymes activate the death pathway. Don't worry too much about death pathways. At this point, we'll talk more about those in clinical reasoning. The chromatin, so the DNA and the nucleus, then starts to break down. Also, the organelles all start to shrink. They start to shrivel up. The plasma membrane, instead of being a nice plump plasma membrane, starts to do this thing called blebbing, where we get the plasma membrane in and out and really uneven, and we've not got a nice shaped cell. But what that leads to, the blebbing, is that the plasma membrane will meet up, it will fuse, and it will fuse around these apoptotic bodies. So everything that was in the cell ends up being sealed separately, admittedly, but it's sealed in plasma membrane. So it's fully enclosed. Any pathogens, any intracellular pathogens are enclosed. Any lysosomal enzymes are enclosed. They can't do any damage within there. So the apoptotic bodies are formed and they contain the cell contents in these small packages sealed by plasma membrane. Ultimately, what will happen, hopefully, in normal circumstances, is that a phagocyte will come along, will bind to these apoptotic bodies, probably via a damage-associated molecular pattern, to a pattern recognition receptor, and will then phagocytose those apoptotic bodies. That will completely destroy anything within there, including any intracellular pathogens or nasty chemicals. In some cases, this doesn't happen. Again, this is something we'll come back to in clinical reasoning when we talk about the pathogenesis of some conditions. If those apoptotic bodies are not cleared away by efferocytosis, the clean up, the phagocytosis of apoptotic bodies, they will eventually degrade. Because if you think about it, they don't have any functional organelles, they have no ability to make ATP, they have no ability to make more proteins to repair themselves. So they will eventually naturally degrade. When they do this, they go into secondary necrosis. And this, we end up with another messy death, because they then release all their contents, we have loads of debris everywhere, just like as if we'd have had lysis. So the ideal is apoptosis followed by efferocytosis, the phagocytosis of apoptotic bodies. That's what we like. Nice clean up. So okay, we've talked about the cells, we've talked about cell death. Let's go for something a little more obvious and visible. And we can look at inflammation and the innate response in terms of the cardinal signs. And most of you could probably relate 3, maybe 4, possibly even the full 5 of these. The first one is heat. Now, where you get local metabolic activity, then you are going to get increased heat. Metabolic activity implies that more ATP is being used, therefore more ATP has to be synthesized. That all creates heat. In this case, of course, we've called a lot of leukocytes out of the bone marrow into the blood, got them into the tissues. They're in this nice local area, and now the neutrophils are throwing out nets. The phagocytes are phagocytosing. All of this takes ATP. It's increased local metabolic activity. That produces local heat. In addition to this, we have increased perfusion. We'll talk about that in terms of redness in a minute. But obviously, if you get an increased perfusion, then that produces a local warmth as well. So redness. Remember we talked about vasoactive amines. We talked about what histamine, bradykinin, prostaglandin, E2 does. They increase vasodilation in a local area. If you have vasodilation in a local area, you're going to get obviously increased perfusion, but also blood is red. So you're going to get increased colour. And in clinical reasoning, we'll talk about circumstances where we get peripheral vasoconstriction, which causes pallor. In this case, we have the opposite. We have redness. As part of those vasoactive changes, we also have increased local vascular permeability. So we have endothelial cells shrinking down, increasing the gaps between them, so that fluid can leak out of the blood, and bearing in mind that most of the blood is plasma and most of the plasma is water, initially this will be transudation. Transudate is protein light. It's thin, watery fluid. Later on, when we start to get cells moving into the tissues and we start to get those proteins that we've talked about, complement, antibodies, that sort of thing, moving into the tissues, then that transudate will become exudate. It will become protein rich. Of course, when this moves into the tissues, it's moving into the interstitial fluid, it's moving into the intracellular spaces. So everything has to move out. Because everything has to move out, we have swelling. Internally, where we have hollow areas, we may get fluid collection. In terms of organs, we might get swollen organs, edematous organs. Edema, posh word for swelling. Those edematous organs make the organs larger. So we get, for example, hepatomegaly, splenomegaly. So we get these organs enlarging because of fluid moving out from their capillary beds into the organs. In terms of tissues, of course, we've got our nice foot there that looks a bit swollen. We can see that. Pain is our fourth cardinal sign. And we have two issues really going on here. If you remember, pain receptors are called nociceptors. They can be stimulated in two ways. Either mechanically by pressure from the edema, or chemically by inflammatory mediators such as bradykinin, histamine, PGE2. Those all bind to receptors on the nociceptors and stimulate them. In addition to this stimulation, they can also be sensitized. So some of those inflammatory mediators, and particularly the neurokinins that we talked about before, like substance P, can sensitize the receptors, the pain receptors on the nociceptors. And when they're sensitized, we will get hyperalgesia, increased pain. So our inflammatory process can not only stimulate pain, it can also sensitize the system, the pains, the sensory pathways, so that they perceive even more pain. And our fifth one is loss of function. In terms of the limbs and joints, this is quite easy to understand, because of course you have a swollen joint, and we've probably all experienced this. You have a swollen joint that's painful. It limits the movement, which is a protective response after all. Let's admit that. It forces us, hopefully, to go and rest it. But in terms of inside us, in the viscera, in the tissues we can't see, we also have loss of function. So potentially things like edema, fluid accumulation, imagine fluid filling up the alveoli. What's going to happen to gaseous exchange? We have cell death potentially within organs, within tissues inside us. And we have attempts at repair. All of these can cause dysfunction of normal cellular activities. So loss of function can be visible, but it also can be invisible. And this is where we get a lot of our pathogenesis from, our pathology of diseases. Okay, so let's look at this all sequentially. We've talked about vasoactive changes and phagocytosis and chemotaxis and all the rest of it. Let's put it together in terms of a sequence. So bearing in mind the innate response can be stimulated by infection or by any tissue damage for whatever the reason. So we could have a sterile inflammation. In other words, infection isn't causing it. Right at the start, remember we said mast cells are key in the early phases. So right at the start, we potentially have mast cell degranulation and secretion. When we get this, we get the release of colony stimulating factors, cytokines, chemokines, and eicosanoids. Remember, our eicosanoids are the products of the arachidonic acid, so it includes prostaglandins. We also, if you remember when we talked about macrophages just now, we talked about tissue macrophages. These are macrophages sitting in the tissues waiting for a problem. So they start phagocytosing any immediate problem, any immediate threat, whether it be dead tissue, whether it be a pathogen. We also may have local dendritic cells doing the same. Of course, when they phagocytose, they secrete colony stimulating factors, cytokines, chemokines, and eicosanoids. So we're building up through the action of a few cells, we're building up this soup of inflammatory mediators that are going to upregulate inflammation. Which brings us to the whole process. So we have partly caused by the histamine from mast cells, but also the cytokines release, lots of other things. We have vasodilation and increased vascular permeability at the site. These are our vasoactive changes. Concurrently with that, we have leukocytosis. So our colony stimulating factors that have been released will release neutrophils in the early stages. Neutrophils are often known as the first responders, although technically I would argue the tissue macrophages are, but never mind. So we will get neutrophilia. Later, we will get a release of monocytes that move into the tissues to become macrophages, and we will get dendritic cells. So we've got vasoactive changes followed by or concurrently with leukocytosis. Both of these together facilitate the emigration of these circulating leukocytes. The emigration involves various processes, including margination, and we'll talk about these processes in detail, adhesion, diapedesis, full emigration, and eventually chemotaxis. Remember, chemotaxis is due to chemokines. Once we have that, then we will have phagocytosis on a massive scale because we've called in all these phagocytes, and that will result in the creation of antigen presenting cells. which is where in the innate response, although this is all still carrying on, we're going to stop, because that creation of the antigen presenting cell is our trigger, it's our step into the adaptive immune response that we'll talk about in the final part. So let's start at the beginning and talk about those vasoactive changes. So let's talk about vasodilation. On the right you can see a normal vessel, so we have our erythrocytes, our white cells, all just moving along in a nice straight line. Now, histamine, bradykinin, prostaglandins, PGE2 in particular, and nitric oxide, remember, is a vasodilator. These are secreted by mast cells or endothelial cells. They bind to the local smooth muscle cells, remember, in the tunica media of the vessel, and cause relaxation in these smooth muscle cells within that area. When they relax, of course, the lumen is going to get bigger. So it widens the lumen of the vessel in a local area. This in turn slows down the blood flow at that point, because it's a bit like having a bulge in a hose pipe. The water under pressure in a nice straight tight line moves quite quickly. It gets to a widened area and it starts to get turbulent and it slows down. The cells within the blood, including the white cells, the leukocytes, start to circulate and fall to the vessel walls. Alongside that, we have increased vasopermeability. Now, whereas in terms of vasodilation, we're talking about the relaxation of smooth muscle cells, here we're talking about the contraction of endothelial cells. So, normal endothelial cells, yes, remember we've got our simple squamous cells forming the tunica intima. They are quite gappy, but not that gappy. Not gappy enough to get a cell between. In this case, again, histamine, prostaglandins, but platelet activating factor, remember this is synthesized after the arachidonic acid cascade from the plasma membrane phospholipids, bradykinin, interleukin-1 and TNF-alpha, all potentially bind to endothelial cells, and this causes them to contract. Of course, when they contract, they're going to leave bigger gaps between them. So we end up with these big gaps between our simple squamous epithelial cells lining the vessels. Alongside that, of course, we have leukocytosis. Leukocytosis is going to release leukocytes from the bone marrow where they're synthesized, and it's going to release them into the circulation. Now, most leukocytes and the endothelial cells can secrete colony stimulating factors. Neutrophils, as we said, these are our first responders, are the first leukocytes that are stimulated by granulocyte, hence the G, colony stimulating factor. They're released from the marrow into the circulation and we get neutrophilia. Later on in the inflammatory or innate process, we get monocytes stimulated. So they're stimulated by granulocyte, monocyte colony stimulating factor, or our macrophage colony stimulating factor. Because of the vasodilation and the slowing of the blood, these leukocytes get to this wider area and they start to fall to the vessel walls. They become turbulent. They start rubbing up against these vasodilated vessel walls. To help them with this and to help them stick to the walls in that area, some of the cytokines and many other mediators increase the expression of something called cell adhesion molecules. Now remember, we talked about these when we talked about plasma membrane proteins back in cell biology. You remember you've got the little two hand-like grips, hooks, hooking each other, one from a cell, one from another cell. And this acts like Velcro. So where we get the increase in expression of cell adhesion molecules on the endothelial cells and on the leukocytes, they're going to grip each other. So we've got these leukocytes in these vasodilated areas going round and round and round because the blood's slowed up just there. They move to the walls and their cell adhesion molecules on them start to grip cell adhesion molecules on the endothelial cells. What's happening is the cell adhesion molecules and the slowing of this blood during vasodilation at this point of vasodilation cause the leukocytes to fall and start to roll along the capillary walls. Now imagine this is like Velcro and you've got something moving at relatively high speed. Initially, it's going to pull away from those hooks and it's going to keep on rolling and rolling until it loses impetus. And those hooks finally make the leukocyte stick to the vessel wall, stick to the endothelial cells of the vessel wall. So eventually, firm adhesion occurs and the leukocytes are stuck to the vessel wall. Once this happens, they can start to emigrate. They're trapped. And because of the increased permeability, because we've got these huge gaps between the endothelial cells, these marginalized leukocytes that are stuck to the vessel walls start to squeeze through the gaps. This starts with diapedesis. This is where they change shape. Using their cytoskeleton, they can change shape, and they put a foot through the gap between the endothelial cells. Eventually, they wriggle all the way through by changing the shape of their cytoskeleton and moving. They can fully migrate through the gap and into the tissue. Once they're into the tissue, you can imagine it. Okay, we've got through the vessel wall. Oh, okay, we're in here. We've been called from the bone marrow. We've been directed to this area by vasoactive changes. We've been trapped by cell adhesion molecules. We've moved through these gaps into the tissues. Now we don't know where to go. So this is where we need chemotaxis. This is where we need something, some signpost to tell them where to go to meet their target. So the chemokines, as they diffuse out from cells in the target area, whether they be macrophages that are phagocytosing or any other cells in that area, as they diffuse out, they form a gradient. This gradient of chemicals, the chemicals diffuse through the intracellular fluid, and of course, closest to the target, they're going to be at the strongest concentration. As they diffuse out, they're going to be weaker, fewer of them dispersed in the interstitial fluid. So they create a bit of a chemotactic gradient. Those chemokines bind to G protein coupled receptors on the leukocytes. So our leukocyte gets through into the tissues and it has a receptor all around it, lots of different receptors for chemokines. And okay, a chemokine binds to one on its nose. It goes, okay, I need to go that direction. And it moves a bit closer. And another one binds to it, a receptor on its nose. It now knows it's going in the right direction. If it moved in the wrong direction, of course, it won't bind any chemokine. Oh, I've gone in the wrong way. So you can see how these chemokines diffusing out leave like a paper trail for our leukocytes to follow into the target. So the location on the plasma membrane of our leukocyte, where the binding occurs, signals to the leukocyte to guide it to the exact site where it is required. Okay, so let's look at leukocytosis again for a start off. We've got in the local area potentially neutrophils, macrophages, mast cells. They secrete colony stimulating factors. The colony stimulating factors bind in the bone marrow and we get leukocytosis. So we get more circulating neutrophils and other granulocytes, monocytes that become macrophages or dendritic cells, and potentially lymphocytes. So in other words, we have leukocytosis. And this is where you come along and you do your blood test, you say, oh, they've got a raised white cell count. And then we have vasoactive changes. The first of these is vasodilation. So the vessel is dilated due to smooth muscle cell relaxation. This slows the blood flow. We also have increased permeability, where the vessel walls are more permeable due to endothelial cell shrinkage. This allows the emigration of leukocytes. So our leukocytes that have been released due to leukocytosis come along, find this widened area, they fall to the vessel walls, they roll, and then they eventually stick to the endothelial cells. This is due to the slowing and turbulence of blood in this vasodilated area and the expression of cell adhesion molecules by both the leukocytes and the endothelial cells. This acts like Velcro to slow and halt the rolling of those leukocytes in this vasodilated area. And then eventually we get firm adhesion. The leukocytes stop rolling. This, as we said, is facilitated by the CAMs. So the cell adhesion molecules have finally slowed them up enough to completely stick to them. And the leukocytes adhere to the endothelial cells of the vessel wall. Once leukocytes are stuck to the vessel wall, they put a foot through the enlarged gaps between the endothelial cells. This is the first stage of emigration known as diapedesis. Ped being foot, putting a foot through. They then squeeze through the gap into the tissues. Once they're into the area, so they're now out of the blood vessel, they're into the tissues. If the leukocyte was a monocyte, it will now become a macrophage. much larger, able to secrete and phagocytose. They know where they need to go because of chemotaxis. So they are directed through the tissues by chemokines. These chemokines are specific chemicals secreted by damaged cells or leukocytes. Chemokines bind to receptors on the leukocytes, and as we said, if they bind to its nose, it knows to follow its nose. If it binds to its other end, then it needs to turn around and go that way. So depending where the receptors are on the leukocyte that binds the chemokine, it will know which way to go. And the chemokines create this chemotactic gradient. The whole process is known as chemotaxis. And as they move through the tissues, just like they did to move through the gaps in the endothelial cells, the leukocytes use their cytoskeletons to change shape and move through the tissues. And this is where we were talking about this increased local metabolism. To do that, they need to use ATP, so therefore they need to synthesize ATP, so therefore they're going to give off heat. Once they are at the site of injury or infection, they can then do whatever their role is. So macrophages and dendritic cells will phagocytose pathogens and the debris of dead host cells. Little neutrophils, of course, will chuck out their nets and release proteases and then quite quickly die. Okay, quick revision on phagocytosis. We talked about it in cell biology, if you remember. We talked about it as a means of endocytosis. So if you remember, what we have is a pathogen-associated molecular pattern or a damage-associated molecular pattern, depending on what the target is, binds to a pattern recognition receptor on the phagocyte. Or, remember, we talked about opsonization. We talked about opsonins, which could be antibodies or could be C3b, one of our complement proteins, binds to the target and then binds to the phagocyte. In the case of complement, it binds to complement receptors. In the case of antibodies, it binds to Fc receptors on the phagocyte. The target is then phagocytosed into a phagosome, a type of vesicle. The phagosome fuses with a lysosome, and this is now called a phagolysosome. Of course, the lysosome is full of toxic chemicals that are going to break down the contents of the phagosome. So the pathogen or debris is broken down by various enzymes, reactive oxygen, proteases, things like that. And the debris is exocytosed. At the same time, when all this is happening, this stimulates the phagocyte to secrete colony-stimulating factors, cytokines, prostaglandins, and chemokines. So lots of pro-inflammatory mediators. This, of course, is going to cause lots of other white cells, leukocytes, to come to the area and come and help it. And this picture was actually sent to me by a student several years ago. It's an electron microscope picture of a phagocyte at work. You can see, whereas we often talk about it phagocytosing one thing at once, actually what happens is that it will phagocytose multiple targets at once. In the terms of dendritic cells, of course, this is a macrophage. In terms of dendritic cells, they have those arms and they can phagocytose lots of things all at the same time. So, as we said, the target binds to the pattern recognition receptor or via an opsonin, such as an antibody, immunoglobulin, or C3b. Pseudopodia start to engulf the target and the receptor. The pseudopodia meet, and the target is completely enclosed in a phagosome. The lysosome of toxic enzymes, proteases, and reactive oxygen moves to fuse with the phagosome to form a phagolysosome. The chemicals from the lysosome destroy the target. The target is broken down into debris, and the debris is exocytosed. Okay, so let's look at a few other associated phenomena, really, in terms of the inflammatory response. One of these is fever. And we're talking about fever in physiology as opposed to pathophysiology because it is a protective response. It has an immunological purpose. It causes the liver and spleen to sequester certain minerals, particularly iron, zinc, and any other minerals. These minerals are needed by pathogens to replicate. So in other words, what it's doing is it's hiding away these minerals so that the pathogens can't use them. Fever also kills or inhibits the replication of some pathogens because they can't survive or replicate within a higher temperature range. It helps to raise our metabolic rate, as does incidentally TNF-alpha. It's one of the things TNF-alpha does. Therefore, what happens is this speeds up the immune response and tissue repair because we now have a higher metabolic rate. All our processes are working at a higher level. It stimulates the release of heat shock proteins. Now, heat shock proteins do have an anti-inflammatory effect, so they help to calm down the response. But an important function of them is that they prevent the cellular proteins from going into denaturation. In other words, remember we talked about how when we synthesize proteins, there can be very complex shapes like channels, receptors, etc. And that shape is key to their function. In high temperatures, they tend to break down. And this is what happens when you fry an egg. The proteins in the white of the egg are denaturing. That's why they go white. They're falling apart because of heat. So in high temperatures, heat shock proteins try and protect the proteins from falling apart. If they can't do that, sometimes they will actually trigger the cell to go into apoptosis, to kill itself, because it can't be protected. Because the metabolic rate rises, fever increases lots of other systems, including heart rate and oxygen consumption, by the rate of about 13% for every 1 degree, if you're not shivering. If you have chills, we'll talk about in a minute, then you're looking at an increase of 100 to 200%. So how do we stimulate fever? Well, fever is stimulated by chemicals that are categorized as pyrogens, not pyogens. They stimulate pus. Pyrogens, pyro as in heat. They can be exogenous or endogenous. So exogenous pyrogens come from, for example, the pathogen. And a main example of this is a protein on the cell wall of many bacteria called lipopolysaccharide, LPS. This can act to stimulate fever. There are also endogenous pyrogens, and this makes sense if you think about it, because we sometimes get fever in sterile conditions, in rheumatoid arthritis, for example. So these are conditions where we don't have an infection, but we still potentially can get fever. This is where the endogenous pyrogens are working on their own. Now, the endogenous pyrogens are all cytokines, and we're mainly talking about interleukin-1 in particular, and interleukin-6. Also, our big bad boy tumour necrosis factor alpha. But all of these, exogenous and endogenous, what they need to do is stimulate the secretion of prostaglandin E2, PGE2, in the hypothalamus, so the other side of the blood-brain barrier. That PGE2 binds to EPR3 receptors in the hypothalamus, and it is only when this happens that we can raise the thermoregulatory centre's set point. We all have a set point. That's our baseline temperature, basically. So all that these pyrogens are doing is raising the set point in the hypothalamus. They are causing the hypothalamus to turn its thermostat up, if you like. As long as PGE2 is bound in the hypothalamus to those EPR3 receptors, the set point will stay raised. Now, we can raise the set point, but of course a lot of the symptoms, the signs and symptoms that we see are due to the body responding to meet this new set point. So fever symptoms are the result of physiological changes stimulated by the set point being raised or being lowered. So you can see here we've got a situation where, first of all, our set point goes up because of our pyrogens. What we then need to do is raise actual body temperature to meet the new set point. And this is where things like shivering and vasoconstriction come in. We'll talk about in a minute. Then we meet the new set point, everything settles down. As soon as PGE2 is not bound to the EPR3 receptors, the set point comes down. So this might be that the whole situation is resolved. It might be that we've taken some ibuprofen or paracetamol and brought it back down because we've got rid of PGE2 and it's no longer bound. So we bring that set point back down. Once we bring the set point back down, of course, the body has to respond by lowering actual temperature to meet the new lower set point. And this is where we see signs such as sweating and vasodilation. So the signs and symptoms of fever are caused by the alterations in actual body temperature to meet the new set point, whether that be higher or lower. So for heat production, what we have is we have shivering due to motor signals. This is our classic chills. This is someone sitting there shivering, saying that they feel so cold, they feel so cold. What that shivering is trying to do is to cause heat to increase the actual body temperature to the new set point. We also get increased metabolic rate. Now, we've already said TNF-alpha increases this, but in terms of fever, fever uses the thyroid hormones to increase the metabolic rate. As we know from when we looked at the thyroid negative feedback loops, the thermal long loop, we know that temperature will rise as a result of an increase in secretion of thyroid hormones T3 and T4. Another way of producing heat is the oxidation of brown fat. Now this is not the fat that we try and get rid of by dieting. This is fat that's tucked away deep inside, primarily in brown places like the kidneys. And it's used for thermogenesis, in other words, creating heat. So when we oxidize it, it releases heat. Alongside that, we can conserve heat. So things that conserve heat are sympathetic piloerection. This doesn't mean that it's being sympathetic. We're talking about the sympathetic nervous system here. The sympathetic nervous system causes the hairs on the skin to stand up. And of course, when the hairs on the skin stand up, it traps heat in so that we have less heat loss. We are conserving heat. Another thing is sympathetic peripheral vasoconstriction. We talked about this in terms of that vasodilation causing redness. Vasoconstriction causes pallor. So the sympathetic nervous system stimulates peripheral vasoconstriction, which of course means that less blood is going to the skin and we become pale. But also we are losing less heat from the blood because it's not going to the skin where it can cool down quickly. Heat conservation, another process, is that huddled body posture. We're stimulated to curl up and try and preserve heat. So these are all signs that what's happening is the pyrogens have increased the set point in the hypothalamus, and the body is trying to increase the actual body temperature to meet that new set point. But when prostaglandin E2, PGE2, is no longer bound to those EPR3 receptors in the hypothalamus, the set point comes back down to our normal. At this point, we have to start losing heat to bring actual body temperature back down to that point. And this is where we see signs and symptoms like sweating, diaphoresis, if you want the posh word. We also see vasodilation causing flushing. So you start to see these signs. It means either the person has taken some paracetamol or ibuprofen, it's got rid of their PGE2 and brought their set point down, or they're getting better. And I don't know about you, but I've seen a few films, very old-fashioned films, where they've said, oh, they're sweating, so the fever has broken. And they weren't wrong, really, because sweating is a sign that, yeah, there's no more PGE2, there's no more pyrogens, so they are getting better. Of course, in these days, it means that we could have taken some antipyretics. Okay, so let's get back on track. So we finished up before fever with phagocytosis. From phagocytosis, it leads into the creation of this antigen presenting cell. And as we said, this is the link really between the innate and the adaptive immune response. So following phagocytosis, normally we've said that the debris is exocytosed, but dendritic cells and macrophages can use that debris and present it to turn themselves into antigen presenting cells. They present the antigen on either MHC2 or MHC1. So this is our little flag of whatever they've phagocytosed, a few amino acids, mounted on our flagpole, our MHC2 or our MHC1. Remember, immune cells have both, whereas all the rest of the cells, the normal non-leukocytes, have MHC1. B cells, the B lymphocytes, they can actually also act as professional antigen presenting cells. We'll look into that more when we look at B cell activation in the next session. So we start with our phagosome, so we have phagocytosis, and of course the antigen that binds is then put into a phagosome. And as in phagocytosis, the phagolysosome forms because we have a lysosome that comes and fuses with the phagosome. And we end up with breakdown of the target. But now what happens is that the macrophage or the dendritic cell will take some of that debris and will mount it on an MHC protein complex on the surface of its plasma membrane. This is now an MHC antigen complex. Remember the little notch in the top of the MHC? This is where our few amino acids of the antigen will be stuck, will be presented. This has now become an antigen presenting cell. Okay, so let's go through it in a bit more detail. So, meanwhile, while we're doing phagocytosis, MHC is synthesized through normal protein synthesis by the phagocyte. It's secured to the inside of a vesicle membrane. So this comes out of the Golgi apparatus as a protein, a structural protein we've made. It's MHC, but it's secured to the vesicle membrane, a bit like exocytosing more channels or more receptors. This vesicle then fuses with the vesicle containing the broken down debris. And a few amino acids of the debris, in other words the antigen, is mounted on the MHC. The MHC antigen complex is moved in the vesicle to the plasma membrane. And we're looking at exocytosis, but we're looking at that type of exocytosis where we keep hold of the protein that we made. We don't release it. What's going to happen is the vesicle will fully open, become part of the plasma membrane, and we've now got the MHC with the antigen mounted in its little notch at the top, sticking out from the outside of the plasma membrane of our phagocyte. It's presented on the membrane of the phagocyte. And as we've said, APCs, antigen presenting cells, are the link between the innate and adaptive immune response. So the antigen presenting cells present this antigen mounted in MHC to lymphocytes, so it needs to go off and find some lymphoid tissue, find a lymphocyte and present it to the lymphocyte. This will happen within lymph glands or lymphoid tissues such as salt or malt. And how they do this is that the antigen presenting cell binds to the lymphocyte via its antigen MHC complex. It binds to the lymphocyte via a specific receptor on that lymphocyte for binding to MHC. We'll talk about more about that in the last part. What this does is it activates the lymphocyte. That lymphocyte will now be specific to the antigen that was presented to it, that was carried to it on MHC by the antigen presenting cell. So that was part 2 of immunology. Exciting stuff, all those cells moving around, our white cells moving around, accumulating, getting into tissues, phagocytosing, etc., etc. Also quite interesting in terms of fever, really, and things that you see in patients are now becoming explained. And it is a defensive physiological mechanism, so we can justify putting it in this bit. We'll come back to it in clinical reasoning, of course, in terms of signs and symptoms. Okay, so we have one part left. In the one part left, we're going to look at the adaptive response. So you can see there we finished off with our antigen presenting cell that's now going to go off and activate the lymphocytes, and we're going to get an adaptive immune response.